So it's my both pleasure and disappointing obligation to say a few words introducing Marvin's thesis. It's my pleasure because he has managed to do something for his thesis that for a very, very long time I thought was impossible. Not anything personal about Marvin, but what I thought was impossible was the idea of finding a way to understand the structure of complex natural acoustic vocalizations, i.e. birdsong, in a way that was tractable enough to both parametrically vary them and generate novel instances of them. And for a long time, people would come into the lab and they would say, well, let's just figure out what the features of birdsong are, and then we'll find out, how the brain sort of maps those features out into the world. And I would say, that's a great idea, why don't you give it a shot? And they would try, and then they would come back to me and we'd figure out what they were actually going to do for their thesis, which wouldn't be that, because that's a really, really hard problem. And so we thought for a very long time about, okay, well, what might these features be? And Marvin came in and he said, well, what if we don't try to find the features, but we just try to find a way that allows us to manipulate these signals. And I think you'll see in the course of the work that he's done, he's figured out a way to get them under control. And then he's used this to understand not only the behavior of how animals perceive these really complex signals, but the neurophysiology that underlies their representation. And so it's a really sort of just beautiful piece of work that he's done that capitalizes on a novel way of conceptualizing the problem and has ended up making a tremendous amount of progress. And so it's my pleasure to introduce him to that. It's also my disappointing obligation to do so, because one of the best, probably the best part about my job is that I get to share my scientific journey with incredibly bright and incredibly talented and really sort of amazing people. And Marvin is all of that. And he's also just an original thinker and a free mind who's brought a kind of life to the lab that is it's special, it's unique, and we're going to miss it terribly. So without further ado, Marvin Till.
Hi, welcome friends. So this quote gives a bit of context to the title of my defense. There's only one thing which is more unreasonable than the unreasonable effectiveness of mathematics in physics, and this is the unreasonable ineffectiveness of mathematics in biology. And this is a reference to an older article that was titled The Unreasonable Effectiveness of Mathematics in Natural Sciences. And since then, this kind of unreasonable effectiveness has kind of been a motif that has been used to talk about how mathematics applies to different types of data. Biology is a little bit more difficult to apply simple mathematics to because of its complexity and the curse of dimensionality. We have 20,000 protein coding genes encoded by 3 billion base pairs in the human genome. We also have tens of billions of neurons and hundreds of trillions of synapses. And they have all sorts of different interactions and emergent properties, so principles don't necessarily generalize from single neurons. Trying to write the equation for how to tell if some pixels are from a hot dog or not would be impossible by hand. However, machine learning allows us to do that. Therefore, the title of my dissertation is The Unreasonable Effectiveness of Machine Learning in Neuroscience, Understanding High-Dimensional Neural Representations with Realistic Synthetic Stimuli, because I highlight some of the ways I use machine learning to compress high-dimensional data into lower-dimensional interpretable representations. The overarching goal of neuroscience, and therefore my PhD, is to understand the brain. And the biological function of the brain is to take information from the external state through perception, do some processing on that information, and then based on that processing, decide which action to choose, which influences the external state, and this loop continuously occurs. My dissertation has been focused on the perception side, where we are trying to understand how the brain processes the information from the external world. Our modern understanding of perception is based on one of the first great discoveries of neuroscience, when Hubel and Wiesel first mapped the receptive field of a primary visual cortex neuron. They found, as illustrated here, that if they move a bar of light into a certain location, the neuron would robustly respond. They then went on to describe how this kind of response pattern could arise by the pooling of activations of several simple cells. Now, this was very promising because if we could do this for every neuron in the brain, we would have a pretty good understanding of how perception in the brain works. This also kind of paved the way for a model where simpler receptive fields were continuously pooled until you got much more complex receptive fields. For example, this is the infamous Jennifer Aniston neuron, where they presented many pictures of Jennifer Aniston, and this neuron robustly responded to these pictures, but not to pictures of other actresses, animals, or places. This is another example of kind of a high-level cortical representation where they showed that there are specific areas that are activated in experienced Pokemon players by images of Pokemon. The primary characteristic of categorical perception is that perception of a stimuli does not smoothly vary with changes in its physical features. That is, in certain situations, small changes in physical properties of a stimuli can cause large changes in the perception of a stimulus. In other situations, large changes can cause no change in the perception at all. We can probably recognize that this is a picture of Jennifer Aniston, and so is this. And we can recognize that these are not the same person, even though it might be closer in physical pixel space. So, in order to explore the neural response around the boundaries of categorical perception, I'm going to explore auditory categorization because neural recordings from cells that are highly specific to celebrity faces are highly regulated and difficult to obtain. Also, auditory categorical perception forms the basis of human language. Every spoken word and phoneme are effortlessly categorized and interpreted. Furthermore, our effort to build artificial text-to-speech and speech-to-text systems have both identified many relevant features where categorical perception occurs. And we've built generative systems capable of constructing a continuum of examples that vary the identified features. Here are the spectrograms of some artificial phonemes that differ only in the curvature of the onset of the second formant frequency. Each equal sign here is a different sound represented as a spectrogram, which shows different frequencies on the y-axis. You can see that the only thing changing between each of these sounds is the front tail on the top part. As I play these sounds to you, you'll notice the perception of three distinct categories: ba, da, and ga. And even though they change by the same amount each time, you'll notice two abrupt changes at category boundaries. So how do we measure categorical perception? If we have two different example categories, Jennifer Aniston and Iggy Pop, or Ba and Da, and we have a way to move between the two categories and create a physical continuum between them, like we heard in the previous example, we can ask someone to classify the stimulus as either A or B. They're going to respond A when near category A and B when near category B. But somewhere in between, you'll find a point where they aren't sure which category it becomes, and they'll respond either category half of the time. This is the category boundary. The sensitivity is the slope of this line, or how steep it is at the category boundary. And this entire curve is called the psychometric function. Alternatively, we can ask someone to differentiate nearby examples on the physical continuum and measure how well they perform. What you find is that people do a much better job identifying these small differences when you're near the category boundary than within a category. And so what this means is that the way we perceive this physical continuum is stretched so that examples near the category boundary seem farther apart, and examples within a category are compressed and perceived as being more similar than they physically are. Another feature of categorical perception, auditory in particular, is that experience influences category assignment. It has been shown that depending on your native language, you will choose to place these boundaries in different locations. So here are two psychometric curves for a native English speaker and a native Spanish speaker, and you can see that where they choose to place the boundary has moved. So categorical perception is an interesting speech phenomena, but in order to get at the underlying neural mechanisms, I'd like to study this in an animal model. The European starling is a good model organism for this because we know a lot about their high-level auditory perception, and they are sensitive to transition probabilities and can recognize patterns that we previously thought only humans could. They are lifetime song learners, so they continuously incorporate new song elements into their repertoire, and every time they sing a new song. They have motifs and song segments that kind of make up their songs, the same way kind of words and phonemes make up our sentences. And avian auditory pallium is homologous to mammalian auditory cortex. And lastly, they're trainable using the behavioral apparatuses that we've used in lab, and we're able to ask them questions about their perception. But to study categorical perception, we need a method to smoothly vary natural signals in a generative parametric manner. We could try to identify features in the same way the few examples of human speech were identified by close inspection of many examples and a bit of intuition. But if we accept not having a specific nameable feature like the curvature of the second formant, we can use the unreasonable effectiveness of machine learning to generate these smooth continuous continuums between any example song segments or arbitrary complex sounds, which could be useful for non-English languages where identification of these features has proven more difficult. So I take 3 billion extracted song segments from recorded birdsong, and I feed them into a deep neural network as a nonlinear dimensionality reduction technique. I take the example song segment, I project it into a low-dimensional representation, and then I train the network to try and reconstruct that song segment as best it can using only the information in this lower dimensional representation. And once I train this on the 3 billion examples, this model then learns a robust representation of these many-dimensional complex stimuli in a much more reduced representation. And so then what I can do is I can take another example stimuli, project it through the encoder of the deep neural network, and get its low-dimensional representation. I can then interpolate between these two representations and then project those interpolations through the decoder to get a smoothly varying continuum between these two example song elements. So this latent space provides a smooth continuum, and so it's illustrated by these numbers on the right here. So these are all examples of morphs that have been created through a neural network in the latent space, and you can see that how much smoother the transformation is than if you try and just do some linear interpolation in the input space. Our lab has also developed other techniques which produce much higher quality examples by explicitly trying to make the interpolations more realistic and encourage convex latent distributions. So for the rest of the talk, we'll talk about two main parts. First, we'll ask the birds what they think about the generated song examples and see how I measure categorical perception across subjects and different contexts. Then we'll measure how the neural activity of secondary auditory regions respond to the song examples and interpret the changes in the neural representations. So first, I ask the birds what they think of these generated song example segments using a simple two alternate choice task. First, the bird pecks in the center port to initiate a trial. I present an example song segment, and they respond either left or right, and if they get it correct, they get a food reward through the hopper at the bottom. Here's an example of the bird doing the task, and he responds correctly, and so he gets a food reward. I start with 8 different example songs, 4 that are associated with a reward for pecking leftward, which I'll call A, B, C, and D, and 4 rightward, E, F, G, and H. Once they learn this task, I can ask them what they think of example song segments from my low-dimensional deep neural network generated space that lie between A and E. I use a ratcheting double staircase procedure to allow the birds to determine their own categorical boundaries. And I can simultaneously do this for all 16 possible interpolated dimensions. So I ask the birds to do this task 5 billion times. So I once again turn to the unreasonable effectiveness of machine learning to make sense of these trials. So what do these trials actually look like? Let's plot some of the trials of a single bird as they respond to song elements that lie between example A on the left and example E on the right. The morph position is the measurement of the physical continuum we discussed in the introduction between these 2 examples. We have trained the bird to respond leftward to example A and rightward to an example E, and lo and behold, it responds strongly to the left when the morph position is close to A, and responds strongly to the right when the morph position is close to E. If we take the average response for bins across the morph position, we can see the characteristic S shape of the psychometric curve. So I once again use machine learning to compress thousands of binary responses over 128 intermediate points into 4 parameters: the category boundary, the sensitivity to changes near the boundary, and 2 parameters which define the absolute accuracy achieved at each of the endpoint samples. But this is just the psychometric function between A and E. I have 16 possible morphs and therefore 16 possible psychometric curves. So I'll be plotting them with the left morph along rows and the right morph along columns. And so there are 16 possible psychometric curves for every single bird that I have behavior from. One thing I can ask is how the threshold and sensitivity varies between each of these morphs. This shows all 16 psychometric functions for a single bird. And as you can see, we find a large amount of variability in where it chooses to place the category boundaries, as well as how sensitive they are to changes along each dimension. This also indicates that our generative model is creating a smooth, diverse stimuli that isn't just flipping in the middle or some other trivial solution to generation. With this variation, we would like to know how different birds respond to the same task. Our subjects are wild-caught animals from LAX, and we have no control over their past experience, and it is known that birds have different regional dialects which could influence their perception in the same way native language influences ours. Or individual birds could just respond in a way based on some random set of preferences they have. But this isn't what we find. So these are the 16 morph dimensions for this single bird again. And if we plot the responses of 3 more birds, we see that these boundaries are much closer than one would expect from random sampling of all psychometric curves, shown again on the left. This indicates a shared perceptual space that drives birds to respond in a similar manner. So what does this mean? We have demonstrated an approach that can take arbitrarily complex auditory examples, generate a smoothly varying physical continuum between them, and ask a bird how they chose to categorize these examples. These perceptual measurements are shared across birds and indicate a shared perceptual space that the birds use to make their decisions. You see the same kind of shared perceptual space in human language with phonomic boundaries, which are, as I've already told you, are experience dependent. So I'd like to see if we can shift the boundaries in my subjects as well. So the way I do that is by training 3 different cohorts. The first I've already shown to you, where I've trained them to classify A, B, C, and D as left, E, F, G, and H as right responses. 2 new birds are trained in cohort 2 with A, B, G, and H as left, and E, F, C, and D as right. And 2 more birds with A, B, E, and F as left, and C, D, G, and H as right. Now these plots are aligned so that it allows us to compare dimensions where different birds are asked to classify the same physical stimuli and associated endpoints where the only difference is the other initial example assignments. Most of these morph dimensions are unaffected by changes in the initial training condition. Here are 4 different birds from cohorts 2 and 3 who all place the boundaries in the same location and have very similar sensitivities. However, on a few dimensions, we see that birds from different cohorts choose to place the boundaries in different locations. So depending on which cohort they're from, they have very different psychometric curves, even though they're still highly conserved within a cohort. So, in summary, using unsupervised machine learning, we can generate morphs between arbitrarily complex stimuli. This works on any animal vocalization or even any language. Birds respond categorically to changes along these morph paths, validating our generative model. Birds respond similarly to other birds, which indicate they have a shared perceptual space. And context and training can cause reliable shifts in boundary locations and sensitivity, further validating our generative model and demonstrating that these curves are not strictly determined by the stimuli, but also depend on the context of the reward associated with other examples. So are there any questions before we move on to the electrophysiology section? All right. So to understand the neural correlates of categorical perception, we're going to be recording from a secondary auditory region called caudomedial mesopallium, or CM, which is known for responding specifically to songbird song, but not to other noises, and is also extremely temporally precise. So 200 hours of recordings later, 200,000 presented stimuli, over 8 birds, and 47 recorded neural populations, and using more machine learning to isolate 2,000 individual neurons that fired 400 billion times, we once again need something unreasonably effective to provide us with interpretable data. I'm going to show the responses of a single CM neuron to these 4 examples, A, B, F, and H, shown in spectrogram on the top and sound pressure waveform below. For a single recorded neuron, I'm going to plot spikes at the corresponding aligned time on the x-axis for 240 presentations of example A, where each presentation is a y value. The dark bands indicate time points where this neuron will reliably fire when I play this example. This neuron also responds in a similarly temporally precise manner to all example song elements, although each has its own unique temporal pattern. So I'm going to convolve a Gaussian with the individual trial spiking patterns to get continuous estimate of instantaneous firing rate for each individual trial. For visualization purposes, I'm going to represent these patterns as a color bar that shows the temporal activations of this single neuron. Then I'm going to look at how these color bars change as we move along the physical continuum from A to F, for example. In this plot, the physical continuum for example A on the bottom to example F on the top is shown. You can see that the top of the plot matches the color bar that occurs during the playback of example F. Example A's response also matches the color bar of the cyan box response. And we can visualize the response of single neurons to changes along individual morph continuum for several different of these continuum. And what this shows is the variety in how firing rates change along these morph continuum. For some, it stops very abruptly at some point in the stimuli. Other places, there's kind of a converging of spike peaks, where 2 spike peaks will become 1 at the other side. And so this is just one of the many neurons I've recorded. So I need a way to compress these representations into a lower dimensional representation. So we create a large table of data where each row represents one of the 4,800 example sample presentations. Columns represent time points of the activations we just showed, concatenated with every neuron we have simultaneously measured, where n is the number of neurons and can go up to possibly 50 neurons at a time. Then we perform logistic regression to classify all trials of example A versus all trials of example E to project this 50n dimensional space into a single dimension which best captures the difference in representation between example A and example E. We do this again for each of the 24 morphed physical continua we have used in this behavioral task. It's important to note that we do this because we have stimuli examples from these morphed continua, but we aren't using any behavioral data to do this. This projects the single trial population activity into 24-dimensional space that contains as much information about the 24 physical continuum dimensions used in the behavioral task. I will call this space Birdnepper for short. This also takes the population activity of different sizes because they have different numbers of neurons and projects it into the same dimension space. So what are we searching for in these spikes? We would like to see how the perceptual continuum, which we are measuring through firing patterns, relates to the physical continuum. So let's go back to looking at how the neural representation changes as we move across a physical continuum. This is the representation we were looking at above, but instead imagine that this is the combined representation of 50 different neurons using Birdnepper. Let's look instead at the difference between representations. If we look at the difference between these 2 points, and we plot the magnitude of the difference between these 2 points as this off-diagonal point here, and since they're pretty similar, we'll plot it in yellow. Now, if we compare the distance of those 2 points to this point, which is quite different, we can see lower similarity. This is called a cross-correlational plot. The larger yellow squares indicate that there are 2 categories where representations within each of the squares are similar to each other but different between the squares. So this is the cross-correlational plot of activity of a single recorded neural population. Over here, we show 2 of these plots where we've averaged across every single neural population we've recorded. And it's not surprising that we see a smoother, less noisy cross-correlation matrix. Although this does demonstrate the relationship between Birdnepper and the physical continuum, it's still hard to interpret. What we would like to know is how much stretching occurs between the physical continuum and the perceptual continuum. So I explicitly define a function along the diagonal of how fast the neural representation changes with respect to changes in the physical continuum. I perform a least squares regression using pairs of neural representations against the integral of this function between the intercepts of the pair to the diagonal. If that didn't make sense, I'm going to use machine learning to estimate how much stretching there is along the physical continuum. If this curve is flat, i.e. the neural representation changes evenly along the physical continuum, we would expect a cross-correlation matrix that looks like this, where the neural similarity is only determined by distance from the diagonal. Instead, we find a curve that looks like this, where there is a peak that's near where the corners of these squares meet. Now, this neural stretching curve depends only on the neural activity from this single population of neurons. However, if we plot the location of the behaviorally determined category boundaries for this dimension, we see that they occur very close to the peak of this curve. Similarly, we can look at the average neural stretching curve for all populations. The peaks of all of the curves occur right near the behaviorally determined category boundaries. And in cases where there's a shift in where the boundary occurs between cohorts, we see 2 peaks in the neural stretching curve. Yes?
So, is this in the same animals? Like, your neural responses are in the same animals that the behavior was done in, or that's a separate set of animals?
So this is actually across some naive birds, some birds that were trained on the task, and some that are. So this is only determined on the electrophysiology. So it's some birds that are trained and some birds that are not. But we can create these curves for stimuli that the bird has never seen, so the dimensions that weren't they didn't do behavior on, and it also works on the naive birds as well.
So the peaks and the neural response space are not necessarily where the behavioral categorical boundaries are established, right? It's not the same animal necessarily.
Yeah.
You're arguing that there's some sort of objective neural representation that results in the behavioral categorical boundaries, but it's not the same animals.
Yes.
Okay.
And so these neural stretching curves capture the structure in these cross-correlation plots. And if you reconstruct the plots using only the neural stretching curves, you can see that most of the structure is actually captured. And as we were just talking about, we can calculate these neural stretching curves for any electrophysiological recording across all 24 dimensions for all of the behavioral birds. And we can see that wherever there's a shift in the boundaries for cohorts, we see 2 peaks in the neural stretching curves, and we also see a lot of reliability in the shape of these neural stretching curves. So to kind of explore how conserved the neural stretching curves are, we're going to try and predict the identity of the morph dimension they come from on held out behavioral data, or held out electrophysiologically recorded data. And so since these are all above chance, which is down here at 1/24, we can see that we can predict which boundary, which morph dimension the neural stretching curve comes from much better than chance. So these are population invariant, or that they don't depend on the neural population that I'm recording from or the bird. Lastly, we use the variation in boundary placement to show that the amount of average neural stretching that occurs at a category boundary location is correlated to sensitivity. So this means that how sensitive the bird is to changes right near the boundary is dependent on, or at least correlated with, how much stretching there is in the average neural stretching curve. So to recap, to the best of my knowledge, this is the first single unit level study where a vast and arbitrarily chosen selection of species-owned vocal communication signals are used to understand categorization. I have created BirdNipper to compare the temporal population representations of arbitrarily sized populations, and I've also developed a method to estimate the stretching of BirdNipper with respect to our physical continuum. I show that behavioral boundaries occur in locations with higher stretching, and dimensions where context and training shifts occur have 2 peaks in the neural stretching curve. I show that these neural stretching curves are the same for different populations, recordings, and birds by predicting the morph dimension they belong to on held out data. And I show that the amplitude of neural stretching that occurs at a categorical boundary is correlated to how sensitive the bird is to changes near that boundary, validating the use of these neural representations as a proxy for the bird's behavioral perceptual space. We also don't find any difference in any effect of training on these neural stretching curves, further strengthening the argument for a shared perceptual space across animals. These results are consistent with the theory that human language developed to use phoneme category boundaries located in regions with intrinsically higher discriminability. Similarly, it's possible that my birds may have chosen to place their behavioral boundaries in a location that had intrinsically higher discriminability to make their tasks easier and they could get more rewards. Lastly, it should come as no surprise that we're able to predict the full behavioral psychometric curves from BirdNipper of a completely naive bird. We train using BirdNipper of a naive bird to try and predict the behavioral curves of one of our behaviorally trained birds and predict a held out dimension. The quality of prediction, of course, varies by population, but we see a significant reduction in error from the shuffled null distribution in purple to the actual data in orange. If we break this up by individual populations and estimate the likelihood of fitting each of these by chance, we once again find no difference between trained birds and naive birds, or any of the other populations of birds versus their own recordings or not. So we can predict behavioral responses using the responses of tens of neurons in anesthetized starlings who have never heard the stimuli before. This is yet more evidence for a shared perceptual space. It also supports the theory that behaviorally, these birds just chose to place the boundary locations with at locations with high intrinsic discriminability without needing to change their underlying representations, just changing how they use them. Lastly, the fact that we can do this with recordings from tens of random neurons pushes back on the idea of an auditory Jennifer Aniston neuron and instead points to a much more dense distribution of information across cells in the auditory pathway. This joins a growing body of work from our lab as well as others, indicating that auditory neurons respond to many features and the brain interprets the activity by integrating activity across the entire population. I think that it is likely that the Jennifer Aniston neuron and even the Pokemon regions likely respond to many other stimuli that just wasn't tested in the limited stimuli set that they were able to present in a human. Lastly, all the analysis and code for this is open sourced, and it runs every time I change any code, and it runs 87% of the code and downloads the data, and it runs in Python 2.7 and 3. So it's very likely that you'll be able to get any of this code to work if you were interested in any of the analysis. So any final questions before I do a quick acknowledgments of some people?